going to have our first talk from Dave Daversa, who will be presenting on applying animal welfare to wild animals. Dr. Daversa is a postdoctoral fellow in the Institute of the Environment and Sustainability at UCLA, where he studies the impacts of stressors on amphibians in California. His foundational research examines stress in the context of wildlife infectious disease, and he has contributed to several publications on the fungal pathogen of amphibians, Batrachochytrium dendrobatidis, which is causing amphibian declines worldwide. His current research examines wildlife stress in the context of human disturbance. And Dr. Daversa uses epigenetic approaches to develop new measures of welfare and stress across animal lifespans. Thank you for joining us today.
Thank you, An, and thank you everyone for joining today. I am going to start here by sharing my screen. Let's see here. Remembering to share the sound. I have a little video clip for you. Okay. All right. Folks can see my screen, I hope. On, yeah. You can see my presentation? Yeah.
Yes.
Okay, good. Awesome. All right, so today I am going to tell you about how we can apply our tools for measuring animal welfare to going out into the wild and studying wild animals. And just a brief kind of introduction. An did a good job of giving my background. I will just do the same. So my name is Dave Daversa. I am a postdoc at the University of California in Los Angeles. I am currently doing some research in conjunction with the Wild Animal Initiative, looking at how we can leverage epigenetic tools to develop new measures of wild animal welfare. All right, so my background, I got my bachelor's degree actually in forestry at Virginia Tech here in the US. And that taught me more than anything that I really liked biology, not so much the industrial forestry side of things. So I went on and continued on to grad school eventually to focus in on biology. So I did my PhD at the University of Cambridge and then a few postdocs both in the UK and the US, and I am currently here in Los Angeles. All right, so broadly speaking, my research looks at the effects of stressors on wild animal populations, and particularly those stressors that are associated with global change. Most of my work has focused on stress in the context of infectious disease. So my PhD and before my PhD, my research was focused on understanding the impacts of this little microscopic fungal parasite, Batrachochytrium dendrobatidis, and its impact on its hosts, which are amphibian species worldwide. We call it BD. BD causes this disease, chytridiomycosis, that is proving detrimental to species, causing mass mortalities of populations and local extirpations. And it's credited with causing more species losses than any known pathogen. So it is one of the preeminent biodiversity threats. More recently, I've kind of expanded my view, thanks in part to the Wild Animal Initiative, to look at multiple stressors, and not just stress in the context of infectious disease, but stress in the context of human disturbance, extreme weather, etc., and also the interactions between these different stressors. All right, so that's my background, and that's what I'm coming to you with today, to talk to you a little bit about measuring welfare and applying these measures to wild animal systems. And I will just say, you know, as my background suggests, I do come from a very much ecological background. And so I do have that bias. And I'm going to talk to you guys a little bit today about, you know, how we approach these topics and how we measure welfare attributes in wild animals from an ecological perspective. All right, so some of the things that would be nice for you all to take away from in these next few minutes is to just become familiar with common physiological measures associated with welfare. I suspect this may be a bit of a review for you that you may have gone over last week. but a review never hurts. But I just want to provide some context and of the measures, go into some details before we start thinking of applying them. Okay, also just considering the advantage and disadvantages of different measures of welfare, learning about the methods that we ecologists use to measure welfare attributes linked to fear and stress. Okay, and then throughout all of this, hoping that you can understand the assumptions and inferences that we make when we attempt to measure and apply um, those measures to understand what welfare is. Okay, so again, just starting with the basics, this is going to be a review for you, but just as a reminder, what we consider welfare to be. Okay, so as Wild Animal Initiative defines it, welfare is the aggregate quality of an individual's subjective experiences over a given time period. Okay, it is a mental state that individuals are in. If we go out and look at how other people have defined welfare, the first thing I tend to do is do a Google search, and that brings us to the Merriam-Webster dictionary. Okay, so how do they define it? They define it as the state of doing well, especially in respect to good fortune, happiness, well-being, or prosperity. Wow, that's pretty broad and probably biased towards humans, but that's how the dictionary would define it broadly. Donald Broom, who you interacted with in the first workshop, would define welfare as an individual state as regards to its attempt to cope with the environment. And I kind of like that one. It's very much resonates with a lot of the ecological literature and how animals face threatening conditions. I'll go into a bit more of that later. And then Harvey 2020 in their paper on 5 domains defined welfare as an animal's mental experiences. In other words, how an animal may be experiencing its own life. Also, again, pretty broad. So we can kind of combine these different definitions to get at kind of what the essence or some commonalities between these definitions to get at the essence of welfare. And I just picked out that first and foremost, this is a mental state of living things. It has to do with individual subjective experiences that they have in this world. And also, there is a link to the environment. Okay, so how animals interact, and we humans, how individuals interact and our relationship with the environment. All right, now, how do you measure this? Yeah, I assume you went through some of this in workshop too, but if we think about these definitions, it really brings us to one of the major problems that we face in welfare science is that we can't get inside the heads of animals. And so to get at the mental state is really challenging. And so I just I just made this little figure kind of inspired by an analogy by Marion Dawkins, just reminding you as I go through everything that I will present today that we can make attempts to measure wild animal welfare, okay? But those measurements, really to a certain degree, no matter what they are, they're going to require some type of inference into how that relates to welfare, okay? Now, does that mean that we shouldn't do it because we can never really get inside the heads of animals? I would argue no. And also, just keep in mind that what we're doing here is we're taking measurements, we're drawing inferences of those measurements to get a sense of what the welfare of animals may be. That's essentially science, okay? Any aspect of science involves a certain degree of inference. And I guess the key is honing our tools, developing and advancing our measures so we can strengthen the inferences we make. And there's a lot of reasons why we should measure animal welfare, right? For practical purposes, traditionally, we do it to identify species for regulation, particularly those that we keep in captivity and do research with. Also, with those animals in captivity and those which we do research with, we can study, we can measure welfare to minimize pain and distress while we're engaging with them. And more recently, and thanks in large part to the Wild Animal Initiative, welfare is going out into the wild and taking measurements of welfare to identify poor welfare, so we can target those populations facing poor welfare to target for interventions related to conservation. All right. So I highlight poor welfare because this does seem to be kind of a bias in the field that we're, positive or good welfare is also important to measure, but there's this bias towards poor welfare. Okay. And so there's some signs that we can look at in measuring poor warfare, or indicators, I would say, of poor welfare. Clearly, there's pain and suffering, okay, how much we are inflicting on animals when we do things like inject them with needles or we put radio collars on, like these people seem to be doing with this elephant here. All right. And also, uh, less kind of, I guess, less concrete signals of poor welfare would be fear and stress. Okay, and these topics I'm going to focus on today because that is more of my background and it's getting a lot of attention by ecologists. So understanding fear and stress and applying that to understand the wild animal systems, both the ecology and the welfare of these wild animals. All right, like I said, in my ecological background, these two correlates of poor welfare have been getting a ton of attention, okay? And so I'm just going to go through some ways that we approach the topic of fear and stress in wild animals. Again, just like welfare, there's a lot of different ways to define fear and stress, okay? So I've highlighted few. Some have said that fear is individual responses to life-threatening events. If you go into the Merriam-Webster dictionary after doing a Google search, this is not specific to wild animals, obviously, but it is an unpleasant emotion caused by the belief that someone or something is dangerous and likely to cause pain. All right. Stankiewicz and Blumstein. Dan Blumstein, by the way, is at UCLA as well. He's just down the hall from me in my office. We're collaborators, and he's written a lot about, and he studied fear a lot. He defines it as the amount of risk animals perceive in a given circumstance. Okay, so I highlight perceive because just to link it back to welfare, the perception of a threat is a mental state. So you can see how this would relate to an animal's welfare. It all goes back to its mental state of perception. And then we don't stop there. We also think, when we think of fear in wild animals, we think of the responses that they are making to that perceived threat. All right, going on to stress. How do we define stress? Well, this is a topic of great controversy because people define it in many different ways. Wikipedia would define it as an organism's response to a stressor. Not very helpful. Begs the question, what is a stressor? Hans Selye, who is known as the father of this topic of research on stress, he would define it as a non-specific response to any demand. Okay, pretty broad. Can we get more specific? Don Bradshaw, a stress physiologist, would define stress as the physiological resultant of demands that exceed an organism's regulatory capacities. Okay, more specific, but perhaps too specific on physiology. And then Robert Sapolsky, whom we'll get to know in a little bit, would define stress as the physical or perceived threat to homeostasis. Again, it's a perceived threat. So we start to see, I throw out all these definitions of welfare, fear, and stress because you start to see some common features, okay? It is a perception, it is a mental state, and it is a state of perceiving a threat to one's well-being. All right, and so how is fear and stress relevant to wild animal welfare? I'll try to get into that more in a bit, but there's a lot of work coming out from the ecology realm that animals that are fearful or stressed out, it just puts individuals in a negative affect. It's not very positive feeling to be fearful and stressed out. And when we are in these states of fear and stress, it leads to suboptimal performance because we just don't work as efficiently towards other essential activities like in the case of wild animals, going out and finding mates to reproduce, or moving around and foraging to nourish themselves. They tend to do that less efficiently when they're in a fearful or stressed out state, and that can lead to compromised welfare. Okay, and just in general, being in this state of fear and stress, it just places a strain on your system. It tends to increase your energetic demand and just the pace of internal functioning that we have, and that and that over time can have detrimental welfare consequences. All right, so just as an example of the relevance of fear and stress to wild animal welfare, there were some folks that went out and studied song sparrows, okay? And they took female adult song sparrows, and with some of them, they exposed them to noises of predators, okay? So, or predator calling. And then with the other group, they did not expose them to predator calling. They did this with adult females, and then they monitored them after they kind of laid a clutch, and they collected their clutch, their offspring, and looked at survival in them in the subsequent years after these females had been exposed to the noise of predators. Okay, so what happened in those offspring, you can see. So the blue dots here in the graph on the right, figure A, the blue dots denote offspring from parents that did not get exposed to predator noises. Okay, and you can contrast that with offspring survival in the red dots, which were offspring that came from parents exposed to predator noises. And you can see over time there is much reduced survival of offspring from those parents that got exposed to predator noises. Okay, so if you take the noise of a predator as a fear stimuli, you can make the indication that that sense of fear reduces survival in these song sparrows. And then they broaden that out to look at the population level effects. Okay, so in figure B here, with the population size on the y-axis, again, the blue dots denoting parents that did not get exposed to predators, that population in subsequent years grew, whereas the population with parents that did get exposed to predators declined, resulting in a twofold differences in the population sizes over 5 years. So fear and stress are relevant to wild animal population viability. All right, so when we go out into the wild, we want to measure fear and stress. There are two different things that we tend to look at. All right, one is their physiology. Two is the behavior of wild animals. I am just going to focus for the purposes of time, unfortunately, on the physiology. I have slides to include to talk about behavior, but I went through them yesterday and it just made all of this way too long. So I am going to focus on physiology. If we have time, we'll get into behavior afterwards. And knowing that you already looked at measures of welfare, I guess I will go through the physiological variables pretty quick. I do get a bit detailed, but perhaps Bonnie will go into more details with you. I will kind of zip through this stuff so we can get at some examples of how we apply this to understand welfare in wild animals. Okay. Again, I'm going to focus on physiology here. All right, and so with these different measures, I just kind of created this figure looking at different axes that we consider when valuing a certain measure for understanding wild animal welfare. It's kind of adapted from Bateson's cube model, but we can think about in terms of its utility and its invasiveness to animals. Okay, we want to minimize invasiveness, maximize the utility of these measures. All right, so just a really brief background.
Pulling him now.
Okay. So just a brief background into stress physiology. Okay, so when we have this perceived threat, it kicks in what's called the HPA axis, which is the hypopituitary adrenal axis, I believe. which essentially starts with the perception of a threat. Okay, happens in your brain, and that triggers different glands in your internal system to work. Your pituitary glands and adrenal glands kick in, and they start releasing different hormones. Okay, such as steroids and adrenaline, and these hormones kind of prime your system to act and alleviate that threat. All right. So a big one when we study wild animals is measuring glucocorticoids. This is one hormone that gets released both in humans, wild animals, captive animals, any type of living organism releases glucocorticoids in when there is a perceived threat. And this is a very common measure that we have for studying stress in wild animals. Okay, but there's a whole range of physiological processes that go on when animals are scared or stressed out. All right, and we can go through and measure any of these if we would like. I'm just going to highlight a few common ones. Okay, so metabolic rate, you can see that happens pretty early on in the stress response. Blood corticosterone or cortisol levels happens a little later. Okay, and fecal corticoid measures. All right. All right, so what happens? Stress physiology 101. And apologies if you already kind of went over this last week, but it's helpful to review this stuff, and it kind of sets the stage for how we apply this stuff in the wild. Okay, so stress physiology 101, hormones. This is a glucocorticoid profile, okay, after a stressor impacts an organism. Okay, so you can think about it with ourselves, with a wild animal that we're studying, whatever. There's a perceived threat, okay, and after a short delay, you see this big spike in glucocorticoid levels, followed by a gradual decline. All right, and so that spike is very typical of a stressor. Just note that there is a little bit of a delay. That's important, followed by a spike and a gradual decline. Okay, so this is what's happening to us physiologically when we are in a state of fear and stress. All right, so we can go out into the wild and measure these things. There's different methods. Perhaps the most common is by collecting blood samples from animals. We can measure glucocorticoids in the plasma section of our blood. So here's a perhaps grim picture of someone collecting blood from the ocular sinus of a lizard. From a welfare standpoint, that seems pretty crazy to do, but studies have actually shown that it's less invasive and less impactful and painful to animals than more traditional approaches, such as injecting them, which you see on the right. Okay. I'm not going to go into the details here about how we measure this stuff. I'm not sure what you all went through already, but feel free to ask questions. I'll give some time for questions here in a bit if you would like more details. We can take that blood, do various analyses to it, measure glucocorticoid levels. Okay, and they do this, we do this with wild animals all the time. Okay, so here is in some great tits in England, they looked at the stress response to a moving owl decoy that they set up in an experimental system of great tits, and they measured this very classic glucocorticoid stress response in them. You see a minor delay followed by a spike in glucocorticoid levels, and then a gradual return to baseline levels. Okay. And I just put this here. They saw that in animals that were exposed to this threat, okay, but they did not see it in other animals that were not exposed to a moving owl. Okay, so applying that to wild systems, we can look at how predators elicit fear and stress with welfare consequences by going out, collecting blood samples shortly after being exposed to that threat and measuring glucocorticoid levels. Okay. I consider this to be for about halfway up on the invasive axis, and it's actually pretty useful because it's very precise and accurate and gives you a good sense of the physiological stress and fear response. All right, you can also measure stress in wild animals through fecal samples, which is less invasive, perhaps not quite as precise. All right, my colleague at UCLA, Dan Blumstein, does a lot of this in his study system of marmots. Okay, so he is very interested in the social dynamics and the social structure of wild marmots in Colorado. He's been tracking them for many, many years. And part of his research incorporates these measures of the glucocorticoid stress response. Okay, so some of his baseline work was just kind of a proof of concept you see on the left hand figure here, with the fecal corticosterone levels on the x-axis and looking at that over time. They injected them with ACTH, which it's a primer for this stress response. Like I said, this was just baseline work. They wanted to see if they could detect a glucocorticoid response, and you can see that they did. It's much more delayed than you get in a blood sample, but that can have some benefits to it, because how do they get the fecal samples? They have to trap these marmots, okay? And they wanted to look at, a stress response, not related to their actual trapping and handling of the marmots, but of another treatment. And so this delayed onset of the glucocorticoid signal in fecal samples can actually be beneficial. And on the right, they applied this measure to look at the motivations behind the alarm calling of animals, these marmots. And they found that those animals that were calling, there was a signature of an increased fecal glucocorticoid concentration, okay, which they used to suggest that alarm calling was associated with a state of fear and stress. All right, looking at it in terms of invasiveness and utility, I would say it's less invasive, but also perhaps less useful than the blood glucocorticoid. This is just my own personal opinion. Feel free to debate me. All right, and a third and final physiological measure is the metabolic rate. All right, we do this through respirometry. I'm not going to go into the details of it now. I actually don't have much experience with this. I hope to kind of get this going in my own research here in the coming years. But metabolic rate, just think of your heartbeat and how much energy you're expending per given unit time. Okay. This is associated with fear and stress. So a lot of people, when they start to think about the welfare of wild animals, they will measure their metabolic rate. Okay, metabolic rate, it does correlate also with other measures of fear and stress in wild animals, such as their baseline glucocorticoid levels. Here measured as cortisol. Cortisol and corticosterone are two types of glucocorticoids. But just keep in mind that it's not always the case that increased metabolic rate is indicative of fear and stress. Here's a study that measured metabolic rates in wild animals, with both that were both nourished with food and starved. And you can see that starvation, which I would argue compromises an individual's welfare, actually reduces the metabolic rate. So we have to think about the context here when measuring these variables. All right, so that is just basic physiology. What does this have to do with welfare? It's a very good question. All right, and so I think I have time to get a sense of why these measures of glucocorticoids and metabolic rate and different physiological variables are useful for understanding the welfare of wild animals. I'll just show you a video here real quick. It's by the great Robert Sapolsky. He applies measures of physiological stress to understand the welfare and the social dynamics of wild baboon populations. And he kind of is very good at elaborating on why these measures are useful in understanding the mental states of animals. All right, let me see if this works.
If you're a normal mammal, what stress is about is 3 minutes of screaming terror on the savannah, after which it's either over with or you're over with.
Over the last 3 decades, Stanford University neurobiologist Robert Sapolsky has been advancing our understanding of stress, how it impacts our bodies, and how our social standing can make us more or less susceptible.
Is the aggregate bad news and
Most of the time, you can find him teaching and researching in the high-achieving, high-stressed world of brain science.
paper is this huge contrast between classical.
But that's only part of his story. For a few weeks every year or so, Sapolsky shifts his lab to a place more than 9,000 miles away, on the plains of the Masai Mara Reserve in Kenya, East Africa.
You live in a place like this, you're a baboon, and you only have to spend about 3 hours a day getting your calories. And if you only have to work 3 hours a day, you got 9 hours of free time every day to devote to making somebody else just miserable. They're not being stressed by lions chasing them all the time. They're being stressed by each other. They're being stressed by social and psychological tumult invented by their own species. They're a perfect model for westernized stress-related disease. Because what stress is about is somebody is very intent on eating you, or you are very intent on eating somebody, and there's an immediate crisis going on.
When you run for your life, basics are all that matter. Lungs work overtime to pump mammoth quantities of oxygen into the bloodstream. The heart races to pump that oxygen throughout the body, so muscles respond instantly.
You need your blood pressure up to deliver that energy. You need to turn off anything that's not essential. Growth, reproduction, you know, you're running for your life. This is no time to ovulate. Tissue repair, all that sort of thing. Do it later if there is a later.
When the zebra escapes, its stress response shuts down. But human beings can't seem to find their off switch.
We turn on the exact same stress response for purely psychological states, thinking about the ozone layer, the taxes coming up, mortality, 30-year mortgages. We turn on the same stress response, and the key difference there is we're not doing it for a real physiological reason, and we're doing it nonstop.
All right, I will end the video there. Yeah, you can go back YouTube Robert Sapolsky on stress if you want to finish it. But I just bring this up because this is Sapolsky's great contribution, and I highlighting that stress is adaptive. It's very beneficial to animals and humans, but but it's also maladaptive. Okay, and how do we distinguish between an adaptive stress response and a maladaptive stress response? It's all about timing. Okay, and so stress responses are supposed to be quick. They're supposed to be short. That short spike in a glucocorticoid levels or metabolic rates, followed by a decline back to baseline levels. All right, that is adaptive. That helps us survive. We need that to happen. Okay, so I would argue that those acute responses may actually be indicative of, over time, positive welfare, because it helps us survive through changing and threatening environmental conditions. Problems arise, the consequences really arise when the stress responses are prolonged, or I would even go further, when they are activated very frequently over time. Okay, so there's a strong temporal component of the links between fear and stress and the welfare of an individual. All right. Okay, here's just a visual illustration of this. So if you measure glucocorticoids in wild animals, if you see a slow return to baseline levels, that's a red flag that, okay, this animal is not really in the best health. The slow return to baseline is associated with, health factors that Sapolsky mentioned as well, such as onset of disease. Okay, so what do we do with this? Or here, I want to show you okay, acute stress versus chronic stress. All right. So Sapolsky argues, and there's a lot of evidence to suggest that it's the chronic fear and stress that really impacts our lives negatively, leading to reduced welfare over time. Okay. And highlighting again that distinguishing the link of fear and stress and the measures that we have, such as glucocorticoids and other physiological variables, the key factor is time. How much these spikes in these stress levels are occurring over time. Okay, so what do we do with that? All right. Well, sorry. We can go out to wild animal populations and we can do longitudinal studies, such as were done with snowshoe hares, looking at their fear and stress when confronted with lynx predators. Okay, the study here did that. They actually did it to kind of try to understand if fear and stress-like states were contributing to the classic population cycles of lynx and hares. All right, they did this through longitudinal sampling of blood glucocorticoid levels. Okay, longitudinal, I mean repeated sampling of a population or set of individuals over time. Okay, we want to get at this sense of timing. All right, but that is pretty laborious, and it's kind of hard to do with many systems. And so I just want to highlight with the last 10 minutes or so here of a measure that I am developing here at UCLA to start to get a sense of fear and stress over longer time spans, particularly over the lifespan of individual animals, because so that we can start to get at these long-term measures of fear and stress and link them to welfare of wild animal populations without having to go through and do these laborious long-term studies. All right. And the approach I'm using is leveraging these new tools in epigenetics. All right, so epigenetics, just the basic definition, we have our DNA sequences, we have our genes, and there's activity going on around our genes that does not alter the composition of our DNA, but it does alter the expression of our genes, okay, into our phenotype. The one that is very popular these days is DNA methylation, and this is what I am using in my research. So we have our DNA sequences inside of us, and over time, our DNA become what's called methylated. These little methyl groups bind to cytosine sites in our DNA, okay, and they accumulate over time, all right, throughout our lives, all right. So some pioneering work done here at UCLA showed that we can measure DNA methylation, and it very strongly correlates with an age of an individual, all right. So it's really a biomarker, a very strong biomarker of aging. They developed this in the health field, but folks like my colleagues here at UCLA are applying it to animals. On the left, you can see how DNA methylation accumulates over the age of domestic and wild dogs. And then some pilot work that my colleague Matteo Pellegrini did in captive bred amphibian xenopus frogs. But this work is still really kind of new, especially in the wild animal realm, okay? So I am currently, along with Wild Animal Initiative, sorry, this is just going through the ways we measure it. I just want to highlight how we're applying these methylation tools, okay? So in collaboration with Wild Animal Initiative, I am looking at DNA methylation in wild populations of Western toads, okay? Sorry, I just want to go back real quick because there's an important point that I need to make, is that DNA methylates as we age, okay? So it's a strong biomarker of aging in both wild animals and humans as well. Now, there's a lot of evidence to show that our DNA becomes methylated faster when we the more fearful and the more stressed out we are. Okay, so if you kind of sample a population of different age classes and look at their DNA methylation, you can compare a kind of this correlation, okay, between age and DNA methylation levels, and populations that are more stressed out or face more fear and stress in their lives hypothetically should have a steeper slope. So DNA should become methylated at faster rates over the lifespan of individuals. All right, so this has been kind of looked at in humans with some validation. Okay, so faster methylation over the lifespan is indicative of higher disease risk and arguably compromised welfare. So I am kind of testing this in Western toads here out in California. All right, so what I'm doing is I'm going out, I am collecting tissue samples of these animals and a little bone sample, so that I can measure their age. And I am looking at this correlation between age and DNA methylation in Western toads. And I am doing that both in non-urban populations outside of Los Angeles. So I don't know if anyone has ever been here, but Los Angeles is blessed in having these beautiful Santa Monica mountains within spitting distance of us. And there's populations of Western toads out there. And there's also populations of Western toads in parks very close to the city. Okay, and so I am collecting samples of these toads, and I'm bringing them back and measuring the DNA methylation levels in toads of different age classes. Right, and by doing that, I am going to compare this relationship between their age and the methylation levels exhibited by these Western toads to test the hypothesis that the urban populations are going to have faster rates of DNA methylation across the lifespan than the non-urban populations, indicating that these animals may be more stressed out and face compromised welfare over time. All right, so I kind of will end it there. That's the main point I want to make, is when we measure these different correlates of welfare, whether it be glucocorticoid levels, DNA methylation levels, the stress response, we want to consider how these measures are affected over the lifespan of individuals and over time in wild animal populations. And I think I am about out of time, so I will end it there and we can go on to questions. I will finish. Actually, I'll finish with a quote by Marion Dawkins. Done a lot of foundational work in animal welfare, and I will let you read it. Just going back to this figure, because I think it's important in any measure of welfare, especially in wild animals, to put the evidence that we have, the data that we collect, in its place and think hardly about how we draw inferences to understand the welfare of wild animals. Thank you.